we would look at the use of light microscopes to observe slides. Now, in preparing slides to observe under a light microscope, the specimen is mounted on the slide. Liquid specimen, example blood sample, is smeared on the glass slide. The specimen is covered with a cover slip. The prepared specimen slide is then clipped over the hole in the stage. A low-power objective lens is first brought above the specimen by rotating the nose piece. The diaphragm is adjusted while looking through the eyepiece to obtain enough bright light. The coarse adjustment knob is turned gradually lower or raise the objective lens while observing the specimen through the eyepiece till a focus of the specimen is obtained. The fine adjustment is then used to obtain a sharp focus of the specimen. There are many methods of preparing temporary slides, but we shall consider a wet mount, dry mount, smear, and squash methods. To prepare a dry mount, which is the most basic technique, select a clean slide. Slice off a thin piece of the sample specimen and position the sliced section on the center of the slide. Place the cover slip over the sample. It is ideal for observing insect legs, feathers, hair, pollen, and dust. To prepare a wet mount requires tweezers, pipette for pipetting liquids, paper towels, and the mounting liquids, which could either be water, brine, glycerin, or immersion oil. Place a drop of fluid in the center of the slide. Position the sample on the liquid using tweezers. At a 45-degree angle, place one side of the cover slip against the slide, making contact with the outer edge of the liquid drop, and lower the cover gently to avoid air bubbles. Remove any excess water with paper towels. To prepare a smear slide, you would require 2 or more flat plain slides, cover slips, pipette, and tissue paper. Pipe a liquid sample, example blood, onto a slide. Using the edge of the second slide, slowly smear the sample to create a thin, even coating. Place a cover slip over the sample, ensuring that air bubbles are not trapped. Remove any excess liquid with tissue paper. To prepare a squash slide, prepare a wet mount as described before. Place the lens tissue over the cover glass and gently press down, careful not to destroy the sample or break the cover glass. Then you squash the sample. You would use tissue paper to remove excess liquid at the end. There are many ways of mounting specialized eukaryotic cells. If a wet mounted or dry mounted slide is pale or colorless, it may be difficult to observe with a microscope. In that case, staining will help to see the shape of each component of the cell, because the components of the cell stain differently with chemicals. To stain eukaryotic cells, we use solutions, example iodine, methylene blue, and crystal violet, which is added to wet or dry mounts. The procedure is as follows. Place a paper towel against one edge of the cover slip. Place a drop of iodine or methylene blue on the other side of the cover slip. Then you wait for the staining agent to be drawn under the slide cover to stain the specimen. You wipe up any excess staining agent with clean paper towel. The resolution of a microscope refers to the ability of the microscope or its lens to distinguish clearly between two objects that lie close to each other as separate points or separate objects, whereas the magnification of a microscope refers to the degree of visual enlargement of an observed specimen. To determine the magnifications of a microscope, we define the total magnification as the eyepiece magnification multiplied by the objective magnification. Example, if the eyepiece magnification is times 10 and the objective magnification is times 40, the total magnification is simply 10 times 40, which is times 400. For biological drawings, the magnification is equal to the size of the image over the size of the object. To measure length using a compound light microscope, we need an eyepiece reticle. It is usually used for distances between 0.2 micrometers to 25 millimeters. The eyepiece reticle is a small piece of glass with a ruler etched into it that fits into a microscopic eyepiece. It could appear as lines, grids, or cross lines. The reticle must always be first calibrated with a stage micrometer. An electron microscope uses a beam of accelerated electrons as the source of illumination, not light as used in the light microscope. The electron microscope produces much more enlarged images with greater details. There are many types. The main types are the transmission electron microscope and the scanning electron microscope. A biological drawing involves making an outline of either the external and internal structure of a biological specimen that is observed. The criteria used to score all biological drawings are the following: a title or heading, the size of the drawing, the proportionality of the image drawn, its magnification, the details shown, the orientation and sections, the labeling and guidelines, and finally, clear and neat lines. We would consider them one at a time. A title is required for all biological drawings. All drawings must be done with a sharp pencil of HB grade. A title may be placed above or below the drawing and must be underlined. Drawings made under microscope must have a name, the section, and the power. Example of a title is a drawing of the foreleg of a cockroach. The size of the drawing must be large enough to cover at least 2/3 of the drawing space. Various parts of the drawing must be proportional to themselves. Example, the antenna of an adult cockroach is about the same or more than the length of the body. The magnification of a drawing is the linear dimension of the image over the linear dimension of the specimen or object. This may be written after the title or under the drawing. Note, it has no unit. Every specimen has specific features that must be shown. Example, the number of wings of an insect. However, for details that are numerous, only a few can be represented. Example, the cilia in Paramecium. Every drawing must be fully labeled. Guidelines or label lines must be drawn horizontally using a ruler. There must be no arrows. Label lines should just touch the features they indicate. They should not be broken lines. Label lines should not crisscross and should be arranged neatly and horizontally around the drawing. Label lines should not carry labels.